Hello everyone, we are going to discuss a new topic, that is the replication in eukaryotics. As you know what mean by the replication is the process by which the exact copy of DNA is get copied from its parental DNA to the daughter DNA, okay, which is occur in the S phase of the cell cycle. That is the synthesis phase, and the location of DNA replication is the nucleus, at which the DNA or nucleic acid is get laid. Okay, well, that is the location of that particular nucleic acid. So DNA replication is the most fundamental process or fundamental action by which um, the information of the DNA is get copied into the daughter strand of DNA. Okay, the producing two identical replicas from one original DNA strand. Okay, the process takes place in the semi-conservative method. So the replication in eukaryotics is as much more complex as that of the replication in the eukaryotics. You have know that the replication in prokaryotics have a enzyme, that is the DNA polymerase. Okay, third, which is responsible for the which is the 10 subunits containing the enzyme which is responsible for the DNA replication. Okay, and there is a initiation, elongation, termination, as same like that. The In the case of eukaryotics there is a three phase, that is the initiation, elongation and termination. What happens in the initiation? In the initiation, there is the origin of replication, at which some specific sequences are present. That sequences is called as the replisomes. Okay, at that point, so the various factors can binds with that origin of replication. Eukaryotic cell have multiple replication sites, as the eukaryotic sequence or eukaryotic DNA, eukaryotic genome is much more complex than that of the prokaryotic case. That's why the eukaryotic cell have a multiple replication sites. Okay, to initiate the replication process, multiple replicative proteins must be assembled on this replication site. So for the initiation purpose, see, there is a various initiative proteins, initiative factors which are binds with that particular proteins that resemble on this replication site. It leads to the formation of replication complex, that is the PRC. Okay, after that what happens for the formation of pre-initiation complex? For that purpose, this is the complex process. So I request to all of you, please take one note pad and or notebook and pen, because this is the complex process which is very hard to understand. So you can take one we can you can take note of this, how the process is get forward one by one. So there is a various factors which comes along in this process. So you can take one notebook and one pen, so you can understand very well. So first, association of origin of replication complex with the replication origin. Okay, so ORC complex is association at the replication of origin. After that, binding of CD6 and protein to the ORC means ORC region complex, that is origin recognizing complex, which recognize the initiation or the replication of origin. At that point, the CD6 protein is get bind after the second step. In third step, there is the binding of CDT1 and many chromosome maintenance protein to that ORC, that is the origin recognized complex. In the this replicative complex assembly occurs during the G1 phase prior to the S phase. That is the these all processes which first, second, third process will get occurred in the G1 phase. Okay, during the transition between the G1 phase to S phase, CDK proteins and DDK proteins get attached to the pre-initiation complex. This is the for the maintain the integrity of that particular genome. Okay, it performs the pre-replication complex into an active replication fork. Okay, this is the diagram. In the G1 phase, there is ORC get binds with the replication initiation site, and there is the CD6 proteins get bind. Okay, in the pre-S phase, that is the CDK protein and DDK protein is get attached with the ORC region. Okay, then the CD6 protein get removed in the S phase, and CDC45 is responsible for the further elongation process. So this is after the initiation there is the elongation phase. In the elongation phase is occurs in the S phase. Then the unwinding of the strand takes place by the helicase activity, which is present in the these factors which are attached with already attached with the initiation phase. Okay, unwinding the takes place by the enzyme helicase that leads to the exposure of the two DNA templates. So for the initiation purpose, for the elongation purpose, there is must unwinding is done by the helicase enzyme. After the unwinding, polymerization of daughter strand takes place. It occurs with the help of DNA polymerase enzyme. So DNA polymerase, which is the most important enzyme in the DNA replication, which involves in the elongation or multiplication of the or copying of the DNA. It involves the in the eukaryotes there is 15 various types of DNA polymerases are there, but three are much more important in the DNA replication in eukaryotic. That is the DNA polymerase alpha, DNA polymerase epsilon, and DNA polymerase delta. What is the feature of DNA polymerase alpha is it is a associate enzyme with the primase, just like it also called as a primase enzyme in the case of eukaryotics, from the RNA primer, which are the 8 to 10 nucleotides long. So main function of the DNA polymerase alpha is to synthesize the primer. Okay, later DNA polymerase alpha elongates the RNA primer by more or 20 nucleotides and then leave the place. Means after the synthesis of primer get attached with the complementary strand at the 3' or 5' end of the DNA. Okay, at that time DNA polymerase alpha can synthesize the particular DNA up to the 20 nucleotides long and dissociate from that place. Then the DNA polymerase epsilon is synthesis of the nucleotide or the addition of nucleotides on the leading strand. Okay, it will continually add the nucleotide at the leading strand to continue process of replication. Thus it will be required only one RNA primer at the beginning. So DNA polymerase epsilon, okay, so require only one type of primer for the initiation purpose. There is a, that means the leading strand synthesis is done by the DNA polymerase epsilon. Okay, then the DNA polymerase delta, it helps the synthesis of the lagging strand, that is mean the Okazaki fragments. It requires number of primers for the repetitively. Okay, so it is a discontinuous synthesis strand. Okay, so make a note that DNA polymerase delta is responsible for the synthesis of Okazaki fragments or the discontinuous synthesis of the DNA in the case of RNA. Okay, this is a diagram of the DNA replication in eukaryotics. You can see the elongation phase. Okay, there is a lagging strand and there is a leading strand. Leading strand involves the continuous synthesis of DNA strand, and lagging strand involves the discontinuous synthesis of DNA. Okay, so you have to draw this diagram in exam. Okay, then the termination process is much more complex in the eukaryotics. Okay, so this problem is solved by the telomerase enzyme. Telomerase is an enzyme which is responsible for the termination process. Okay, so it extend the telomerase extend the 3' end of the parental strand beyond the 5' end of the primer's daughter strand. Okay, so telomerase form RNA complementary base pairs. The process continues. Then the primase and DNA polymerase delta help the further replication. You can see in the diagrammatic representation there is a telomerase enzyme which synthesizes continuously parental strand from the 5' to 3' end. Okay, so associated with the RNA that complements, that is the 3' overhangs. Okay, at the chromosome. So there is a addition of various RNA at the 3' end. So there is a termination get occur and the process of DNA replication get stopped. Okay, thank you very much.